We're going to be doing the DNA extraction from a kiwi experiment. To gather the materials together, we need a kiwi, a knife to cut it up. We have 2 beakers here that we're going to use. You could use a plastic cup if you wanted to. A fork to smash it. We have salt, shampoo, a test tube, a small pipette, coffee filter, denatured alcohol, distilled water, and a graduated cylinder. We also have our safety equipment, so I'm joined by my lovely assistant here. We have our safety goggles because we will be working with denatured alcohol, and that could be dangerous for the skin and the eyes. I also like to wear an apron because this can be messy. Okay, what we're going to be doing is extracting the DNA from the plant cells inside of the kiwi fruit. In order to do that, what we're going to do is we're going to go step by step. What I'm going to do is take the knife and I am going to slice thin slices of the kiwi. You could get about 2 or 3 thin slices. What I'm going to do is place them in one of the beakers or cups. You can remove the skin because the skin doesn't have the plant cells that we're looking for. So I put in 3 slices. Then what we're going to do is measure 30 milliliters of distilled water. I'm going to use the graduated cylinder. I'm going to add the 30 milliliters to the beaker with the kiwi in it. After that, we're going to add a pinch of salt and some of the shampoo. Now, the reason that we're doing all of this is that we're trying to isolate the DNA that's inside of each of the cells. So, first step is to add the water. Then I'm going to have my assistant here start to squish it up with the fork. Okay. So give that a go. So each of these cells has DNA in it. Our goal is to break the cells apart, isolate the DNA, and be able to get the DNA where we can see it separately from the rest of the cell. Okay, now I'm going to add a pinch of salt. It's not an exact amount that's really necessary. Just a little bit like that, a little shake works just fine. You can pulverize it, keep crushing it up. And we're going to take the shampoo. It's difficult to measure the shampoo inside of the graduated cylinder because it's sticky and it doesn't pour out well. So we've measured approximately 10 seconds of squeezing this bottle will give us the desired amount. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. In order to do this experiment, you need to use clear shampoo without conditioner in it. So let me see this. What we're going to do is get this nice and mashed up, pulverized where the fruit is completely squished and the mixture has been around a bunch of times. There's going to be a little bit of bubbles forming. You could stop when you see the bubbles forming. We're now ready for the next step. We're going to take the coffee filter and we're going to take the other container, secure it with the rubber band. Well, this part is important. You need to push that down so there's a little bit of a depression in there so that when you pour the mixture in, it has somewhere to sit and filter through. So if you want to pour that into there, all of it, okay. This is where it can get a little messy, but that's fine. That's fine. That's fine like that. You need to let it sit for a few minutes now. So while that's filtering, I'll go through and explain what each of the ingredients in this mixture is for. Now, like I said, we're trying to extract DNA. That's our ultimate goal for this experiment. So what we're doing is we need to get rid of everything else that's in the plant cell. And anything that's large will be caught and will not pass through the filter. So we use the shampoo because it dissolves the fatty cell membranes. Each cell is surrounded by the phospholipid bilayer. This breaks that up. This bursts the cell, causes all of the organelles to be free and move around. When you squish it up and you add the water, you're increasing the surface area and you're increasing the amount of the kiwi fruit that's going to have access to the mixture. The salt that we added causes the proteins and the carbohydrates to participate. So what we're doing is between the soap or the shampoo and the salt, we're breaking apart the cells and we're gathering all of the parts that we don't need are coming together. That way it's easy to filter out. So then what filters is going to be mostly the separated DNA. Now, the next step is going to involve using the denatured alcohol. Denatured alcohol can be hazardous to your eyes and your skin, so you need to be careful when using it. So I am going to measure, you can hold that for me. I'm going to measure out 10 milliliters of the denatured alcohol. And very carefully, I'm going to pour it into the test tube. Okay, you can hold that. Now when you hold the test tube, hold it like this so that we can see the actual results on the bottom. Okay, now this had a few minutes to filter through. So we don't really need that much of this solution. So what we're going to do is take the filter off, and that what's left, the liquid on the bottom, is what we've been trying to get. Save the rubber band. Now, I am going to slowly take the pipette, scoop up 1 pipette's worth of the liquid that passed through the filter, and slowly squeeze it into the test tube. Now, this is filled with denatured alcohol. The DNA doesn't dissolve in the alcohol solution, and it kind of coagulates to make it kind of looks like jelly right there where the top is going to be. The top of the alcohol is right now. So all the DNA is going to collect there, and you'll be able to move it around and you'll be able to see bubbles forming on it. So I'm going to go ahead and do that now. Scooping up 1 pipette's worth. Slowly pouring it in. And now, you can see, what we're left with is the isolated DNA from the kiwi fruit. And you can see it's kind of hard to see through the camera, I'm sure, but you can see that there's bubbles forming around each different strand of DNA. And you can actually spool it together. You see that, Robin? You can actually spool it together and pull out strands of DNA. So it's very clear to see that we have isolated the DNA, and that you can actually move it around and manipulate it with the pipette. Now this is a great demonstration to illustrate how to extract the DNA, and it has many applications in the real world, such as DNA fingerprinting and all types of biotechnology sciences. This is the basic first step, is isolating the DNA. So basically what you have there is coagulated on the bottom. I'll put it against my dark right there. So maybe that's a little easier to see. You can see that that is the isolated DNA. All right, super.